I work with Drosophila melanogaster, fruit flies, that you might see buzzing around in your kitchen over the bananas, and you shouldn't squash them. And here's why. I've just been awarded a fellowship to study spinal cord injuries in this model organism. Now, fruit flies are invertebrates, which means that they don't have a bony vertebrate column, but they do have the equivalent of the spinal cords, the nerves and whatnot that go within the spinal column, and that's called the ventral nerve cord. And the question I want to ask is, if I take embryos and I line them up and I wound the spinal cord or the ventral nerve cord with a glass needle at the end of development, when most things are done and they're ready to hatch, what can I see? What genes are turned on? What genes are turned off? What changes occur in this organism? And can I use the data that I get to better understand the effects of wounding the spinal cord, so spinal cord trauma, in vertebrates like humans? I work with flies because they have a sequenced genome. We know where the genes are, and to some extent we know what all those genes do. Flies are related to humans. We share about 70% of our disease-causing genes with them. So to better understand a disease in humans, you might want to start with flies because, as I said, they have a sequenced genome, they have a very short lifespan, and really importantly, there's lots of genetic and molecular tricks that you can use to look at functions of genes and proteins and RNAs within that organism. So I've been awarded this fellowship to do not basic research, but essential research. This work, hopefully, will help us better understand the effects of trauma on the nervous system. And looking at the data set that comes out, perhaps we can better devise methodologies of studying these genes in now model organisms that have a vertebral column, and then perhaps onto humans.